Dear students, next topic is lipid metabolism. In this session, we are going to discuss about beta oxidation of fatty acids, ketogenesis and their regulation. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Dear students, lipids are universal biological molecules. Not only does this broad class of compounds represent the primary structural component of biological membranes in all organisms. Among these, lipid can be metabolized by microbes for use as a primary energy source. Fatty acids are the building blocks of lipids that you know. Fatty acids are made of a hydrocarbon chain of variable length that terminates with a carboxylic group, that is the COOH group. The fatty acid structure is one of the most fundamental categories of biological lipids. It is commonly used as a building block of more structurally complex lipids such as phospholipids and triglycerides. When metabolized, fatty acids yield large quantities of ATP, which is why these molecules are important energy sources. Lipids are an energy and carbon source. Before complex lipids can be used to produce energy, they must first be hydrolyzed. This requires the activity of hydrolytic enzymes called lipases, which release fatty acids from derivatives such as phospholipids. These fatty acids can then enter a dedicated pathway that promote stepwise lipid processing that ultimately yields acetyl coenzyme A critical metabolite that conveys carbon atoms to the TCA, that is tricarboxylic acid cycle, to be oxidized for energy production. In addition to their role as the primary component of cell membrane, lipids can be metabolized for use as a primary energy source. Dear students, in this session, we can discuss about lipid metabolism. Lipid metabolism involves the degradation of fatty acids, which are fundamental biological molecules and the building blocks of more structural complex lipids. In order to be metabolized by the cell, lipids are hydrolyzed to yield free fatty acids that then converted to acetyl coenzyme A through the beta oxidation pathway. We can discuss in detail about beta oxidation pathway, the bioenergetics and regulation of fatty acid metabolism. You know, fatty acids are an important source of energy. Oxidation is the process where energy is produced by degradation of fatty acids. There are several types of fatty acids oxidation like beta oxidation of fatty acid, alpha oxidation of fatty acids and omega oxidation of fatty acids. Beta oxidation is the process by which fatty acids in the form of acyl coenzyme A molecules are broken down in mitochondria or in peroxisomes to generate acetyl coenzyme A that enters TCA cycle. It occurs in many tissues including liver, kidney and heart. Fatty acids oxidation doesn't occur in the brain as fatty acid can't be taken up by that organ. There are three stages of beta oxidation. First one is activation of fatty acids in the cytosol. Second, transport of activated fatty acid into mitochondria, that is carnitine shuttle. And the third is beta oxidation proper in the mitochondrial matrix. First one is activation of fatty acid. This proceeds by fatty acid thiokinase or acyl coenzyme A synthetase present in cytosol. Thiokinase requires ATP, CoOH, Mg2 plus. The product of this reaction is fatty acid acyl coenzyme A and water, that is shown in this figure. Second step is transport of fatty acyl coenzyme A from cytosol to mitochondria. Long chain acyl coenzyme A traverses the inner mitochondrial membrane with a special transport protein mechanism called carnitine shuttle. See, this is the carnitine shuttle. Here, this fatty acid transport from cytosol to the matrix of mitochondria. This diagrammatic representation shows fatty acid which enter inside the cell, then to cytosol, then enters into the inner mitochondrial membrane, that is a mitochondrial matrix, then enters beta oxidation, TCA cycle, and finally go to electron transport chain. Next is transport of acyl coenzyme A into mitochondria. Acyl groups from acyl coenzyme A is transferred to carnitine to form acyl carnitine catalyzed by carnitine acyl transferase 1 in the outer mitochondrial membrane. Acyl carnitine is then shuttled across the inner mitochondrial membrane by a translocase enzyme. The acyl group is transferred back to coenzyme A in matrix by carnitine acyl transferase 2. Finally, carnitine is returned to the cytosolic site by translocase in exchange for an incoming acyl carnitine. Actually, these are shown in the previous figure. Third one, beta oxidation in mitochondrial matrix. There are 4 steps involved. Step 1, oxidation by FAD linked dehydrogenase. Step 2, hydration by hydratase. Step 3, oxidation by NAD linked dehydrogenase. Step 4, thiolytic cleavage by thiolase. In the first reaction, the oxidation of acyl coenzyme A by an acyl coenzyme A dehydrogenase to give alpha beta unsaturated acyl coenzyme A, otherwise called enoyl coenzyme A. FAD is the hydrogen acceptor. So that is shown in this diagram. The second reaction is the hydration of the double bond to beta hydroxy acyl coenzyme A or para hydroxy acyl coenzyme A. Third reaction is the oxidation of beta hydroxy acyl coenzyme A to produce beta keto acyl coenzyme A, NAD dependent reaction. The fourth reaction is the cleavage of the 2 carbon fragment by splitting the bond between alpha and beta carbon by thiolase enzyme. That 4 steps are shown here. Here is the energetics of fatty acid metabolism. That is, FADH2 will give 1.5 ATP, NADH2 will give 2.5 ATP, each cycle 4 ATP. Palmitic acid 7 cycle, 7 into 4 28. Acetyl coenzyme A 8 into 10 ATP 80. Activation energy loss 2 ATP. Net energy 108 minus 2, that is 106 ATP molecules. That means a large amount of energy is released during the catabolism of fatty acid. The beta oxidation is regulated by the availability of fatty acid influences beta oxidation. Glucagon, by activating hormone sensitive lipase, increase free fatty acid level in blood. Insulin inhibits beta oxidation by inhibiting this enzyme. Malonyl coenzyme A inhibits catalase 1 activity. Another part is ketogenesis. Acetoacetate, beta hydroxybutyrate and acetone are the ketone bodies. This take place in the liver mitochondria. Step 1, 2 molecules of acetyl coenzyme A condense to form acetyl coenzyme A by thiolase or acetyl coenzyme A synthetase. Step 2, acetoacetyl coenzyme A condenses with another molecule of acetyl coenzyme A to form 3-hydroxy-3-methyl glutaryl coenzyme A, otherwise called HMG-CoA by HMG-CoA synthetase enzyme. Step 3, HMG-CoA lyase cleave HMG-CoA to acetoacetate and acetyl coenzyme A. In the fourth step, acetoacetate is the primary ketone body. It is reduced to beta hydroxybutyrate by beta hydroxybutyrate dehydrogenase using NADH plus H plus as coenzyme. Acetoacetate undergo non-enzymatic spontaneous decarboxylation to acetone. Next, we can see the fate of ketone bodies. 3-hydroxybutyrate is the predominant ketone body present in blood and urine in ketosis. Liver cannot utilize ketone bodies. It lacks the particular enzyme, the coenzyme A transferase or thiophorase. Peripheral tissues utilize them. Succinyl coenzyme A, acetoacetate coenzyme A transferase or thiophorase. Succinyl coenzyme A plus acetoacetate gives succinate plus acetacetyl coenzyme A. The regulation of ketogenesis include, if there is an increase of lipolysis, there is increase of ketogenesis. Insulin inhibits ketogenesis. Glucagon and norepinephrine promotes. In diabetes mellitus, due to insulin deficiency, ketosis occurs. Starvation increases ketogenesis. Dear students, from this discussion, I hope you could understand what is lipid metabolism and what are the pathways fatty acids undergo degradation and energy is released. Thank you for watching. Don't forget to leave a like. and please subscribe my channel.